Okay guys, so last time we talked about osmosis. We went over the terms hypertonic, hypotonic, and isotonic solutions. Then we started to go over maintaining homeostasis with respect to the paramecium and it living in a hypotonic environment, being a single-celled organism or unicellular, and it being it needing that contractile vacuole to make sure that it can regulate its water intake so that it does not go through cytolysis, in other words, get real big and explode. So, other part of homeostasis here. So eukaryotic cells, they can also function as hypotonic environment where we might have an influx of water because we would be hypertonic to that hypotonic environment. Okay. However, our vacuoles, we don't have contractile vacuoles, us and plants. Okay, so it's a little bit different for us. So we want our solute particles to reach equilibrium, guys, just like that. For plant cells, all right, it's a little bit different. So the plant cells actually function best in a hypotonic environment, meaning the cells would get big. So the cells would swell. They would have an intake of water. Why this is because the vacuole would be nice and filled. Okay, it'd be saturated with water. And what that does is the vacuole presses up against the cell wall. And that keeps the cell nice, plump, and square-like, I guess you could say. So if it is like this, if the plant is in a hypotonic environment, meaning the cell is hypertonic to the environment, this will keep the cell from wilting. It'll stay nice and plump. And when the plants are in an isotonic, or even worse than that, a hypertonic environment, the vacuole will probably not have a whole lot of water in it, and the plant will wilt. Okay. What we call this is turgor pressure. That is the pressure that is exerted from the plant and from the water molecules against the cell wall. So you guys can see here, the plant on the left would be in a very nice hypotonic environment. The one on the right, either isotonic or the opposite, hypertonic environment. So our next one we're going to go over is called facilitated diffusion. So we already talked about diffusion. Diffusion moves small nonpolar molecules right through the phospholipids. We then went over osmosis, which is when they are too big. The molecules, the solute particles are too big. And instead of moving those, we had to move the solvent. Now we're going to go over facilitated diffusion. And what we're going to do with facilitated diffusion is we are going to now move those bigger solute particles. In this example, they are going to be large polar molecules. It's still going to move high to low concentration just like regular diffusion. However, the difference between them is that we are going to move them through a protein. And there are two different types of proteins we can move through. Whoops. The first one is called a channel protein. It's like a tunnel, okay? We can get whatever it is, these larger polar molecules, through one side of the cell membrane to the other side through this channel protein. And this is what they look like right here. Okay. The other one is called a carrier protein. All right. Now, what the carrier protein does is it almost acts, I don't know, kind of like a passageway, but it's blocked off at one side. So it might be open like this. Particle drops in. Once the particle drops in, it closes on this side, engulfing the particle, and then it opens up on the opposite side. So it definitely has a route in which it needs to go through this carrier protein. Okay. All right, and here's the second part of it, guys, that I just talked about, it changing its shape. Okay, so it opens on one side and then closes and opens on the other side. All right. Okay, and here's what they look like, guys. There is that opening and closing on both sides. So things that are going to move by facilitated diffusion, the big one is glucose. Glucose, larger molecule, C6H12O6, It's also polar. It combines with water. It dissolves in water. Okay, so it is too big to fit through the phospholipids. So we are going to have to find a protein that can move glucose. And how we find that protein is each of our proteins are going to have a carbohydrate tag on the one side. And that carbohydrate tag is going to allow the glucose to find it and be like, oh, hey, this is the protein I need to use to get to the inside of the cell membrane. Okay? All right. So here's some examples for you guys. If this one, if you look here, we are moving through a protein. In this example, it's a carrier protein because it's opening and closing, right? This one would be facilitated diffusion because it is moving through that protein to one side to the other, okay? This one, if you see the water level dropped, this one would be osmosis, okay? This one, if you look closely, okay, this one's kind of weird, right? You got to determine what is moving in here, okay? Now, something is moving. It kind of looks like water, so you're like, oh, yeah, it might be osmosis, but look what it's moving through. Water has to move through an aquaporin. It is not moving through a protein. Instead, it's moving right through the cell membrane. So this one, since it's moving right through those phospholipids, this one would be diffusion. Okay. All right. Start a little bit of active transport, and then we'll call it a day. So we are done with passive transport now. We went through diffusion, facilitated diffusion, and osmosis. Now we got to go through active transport. So what we're going to do with active transport is we are going to move the opposite way. Diffusion, osmosis, facilitated diffusion. We are always moving from high to low concentration. Active transport, we are going to go against the concentration gradient. What this means is we are still moving those solute particles, but we are going from low to high concentration. Okay? And let me get these lights back on. All right. So again, guys, we're moving from low to high concentration, still moving the solute. Because of this, because we're moving against the concentration gradient, we're moving from low to high. We need energy, we need ATP in order to accomplish this goal. All right. Passive transport, it's like throwing a rock down a hill. No energy is needed, it's very easy. Active transport, oh, look, this is hard. No. Active transport, it's like moving it up a hill, okay? It's definitely going to require energy, a little more work. All right. Okay, some examples of this. Cell getting rid of waste, moving nutrients in and out of the cell. The big one, the main one for active transport is going to be something called the sodium-potassium pump, which we're going to go over. All right. Gatorade. Let's talk about Gatorade. So what is Gatorade? Have you ever tasted Gatorade? It's got a little nice sweet taste to it, but is Gatorade really sweet? No, it's not. Why do you drink Gatorade? Well, people drink Gatorade to quote unquote restore their electrolytes. All right, fancy term there. What are electrolytes? All Gatorade is, guys, is sweet salt water. That's it. Okay. Wherever salt goes in your body, water will follow. So the goal of Gatorade is to get salt into your cell. That's the electrolyte. And so if salt is in your cell, the water is going to follow, and it'll keep you from becoming dehydrated because it's filling up your cells with water. Okay? So do we want sodium in our cells, though, in large quantities? No, because if we got a lot of sodium inside our cell's going to get so big and it's going to explode and the cell's dead now, and that's not good. So we want to get sodium outside of the cell. We want potassium to go in. Sodium is Na on the periodic table. Potassium is K. All right. So we want sodium to leave, potassium to enter. Why we do both of them? Sodium and potassium both have a positive charge. We want to try to get that charge across the cell membrane fairly equal, okay? Fairly equal. We want the cell membrane to be a little bit charged on one side compared to the other side, but We want it to be fairly close to neutral. So here is the sodium-potassium pump. What we are going to do is originally, we do not have a whole lot of sodium inside the cell, but we want very, very little because that sodium inside the cell is going to cause the cell to get bigger. So what we're going to do is we're going to take 3 sodiums from inside the cell and we are going to put them over to the sodium-potassium pump and pump them into the extracellular environment outside of the cell. In doing this, we are also going to bring 2 Ks into the cell. So 3 NAs go out, 2 Ks go in. In order for this to happen, that sodium-potassium pump needs a phosphate, which it gets from ATP. ATP in turn gets transformed into ADP, all right? And that's the whole sodium-potassium pump. So 3 sodiums leave, 2 potassiums come in, all right? Okay. So that is all your notes, guys, on what we just went over. 3 NAs out, 2 K pluses in. We're going to use both carrier proteins and channel proteins in active transport. So here's another example, guys, of active transport. We have very little solute inside the cell, and we're moving it to the, well, I should say on the left side here. We have very little on the left side. We have a lot on the right side. We are pumping it from left to right, so it's going against its concentration gradient. All right. Okay, so I think you guys are pretty good on that. All right. So next thing, guys, we're going to go over how vesicles are used. Now, sometimes it's really not going from high to low or from low to high. If you're only moving like one thing, you really can't justify it as being high to low or low to high. But there are 2 different mechanisms called endocytosis and exocytosis. Very simple. Endo means in, exo means out. Endocytosis move things into the cell, exocytosis move things out of the cell. Both of these are going to move very, very large things, so they're both going to require energy. Yeah. So endocytosis, we might be moving fluids, we might be moving macromolecules, something large into the cell. How we do this is we are going to take a vesicle. If you remember the vesicle, we used that for proteins before to transport the protein throughout the cell. We are going to take this vesicle and push it against the cell membrane or whatever we're transporting. We're going to push it against the cell membrane. Let's say it's a solute particle. Push it against the cell membrane. Okay, it'd be a very big solute molecule. And as it pushes against the cell membrane is going to cave in and almost form a pocket around whatever the molecule is we're trying to get into our cell, and then it's going to close on it. The cell membrane will then reform, and there will be a vesicle pinching off from there, kind of like this. You guys can see that the cell membrane was originally straight here. Solute particles come in, we get a bunch of them, the cell membrane caves in on itself, pinches off, and then the cell membrane will reform on top. That is endocytosis. All right. Okay, guys, I think that's enough for today. We will finish the chapter next time. Have a good day.